Alright, welcome back. Hopefully you've colored it in correctly. So it should look like this. So every T and A go together, A and T. C and G, G and C. T's are green, A's are red, G's are orange, C's are blue. Okay? So the other thing you need to do before we start working on the inside is you're just going to kind of draw a little bond. So these two are bonded together. Okay, so they're kind of holding hands in the middle. And it's important, so I'm going to circle one of the bonds in the middle. I'm going to draw an arrow over here. And it's important that you know that these bonds are called hydrogen bonds. So these are joined by hydrogen bonds. Now the reason this is important is because hydrogen bonds are super weak. It's kind of like Velcro. Like if you were gonna like hang something really heavy from the wall, like a 40-pound weight, you're probably not gonna wanna stick it to the wall like with Velcro because it's not going to hold it up very well. What's good about having weak bonds though for DNA is that it's really easy to break it down the middle and then put it back together, which is going to be important as you'll see in a minute. Okay, so in the middle here they're joined by hydrogen bonds and they're very weak and easily broken, kind of like Velcro. Okay, so what we're going to do is you're going to open up your foldable and then you're going to draw a line. So here's the fold right here. You're going to leave maybe about a finger's width of room and just draw a line straight down because we need to leave some room to write some letters in, okay? And then same on this side, so about a finger's width of room, and you're just gonna draw a line straight down. Use a ruler if you really have to, but look, I did it, no ruler. Perfect, it's perfect, okay. So now we have our lines drawn. We are going to title this S phase, DNA replication. All right, so now if we look at our front again, this is our single strand of DNA, okay? DNA is double-sided, so there's one side, two sides, and it's twisted like a ladder called a helix. This is a double helix because there's two of them. And we're gonna label, this is going to say original DNA on that side and on this side. It's going to be important that it's on both sides. Alright, so now we have this labeled as our original DNA. So what S phase does is it makes another copy because every cell has to have the same amount of DNA. And if you just kept dividing DNA in half, eventually you would get down to none. And that's really bad for you. So it needs to make a copy so that when it divides, one copy goes to one house cell, like a house, and then one copy goes to the other cell. Everyone's happy, everyone has all the same stuff, no one gets mad, it's really great, okay? So we're gonna write that the DNA needs to make, I'm sorry, this is so squishy, one extra copy to go into the new cell. So this would be like if you were going to own another house, you would not just take some of your stuff and put it into your new house, okay? That wouldn't work. If you were going to live in both houses, you couldn't just have half a bed, and you couldn't just have like a washer and no dryer, or you couldn't just have half a stove. Okay, you're going to need all of them. So you're going to have two beds, one in your new house, one in your old house. You're going to have a whole stove, one in your new house, one in your old house. Okay, hopefully you have a washer and dryer if you're lucky. You can have washer and dryer in one, washer and dryer in the other one. So you don't have to split between things, okay? Well, that's what happens here too. We're going to make an extra copy, and the way that it does this is there's an enzyme called DNA Helicase, which hopefully you saw from the posters around the room. So step 1, It's really like a two-step process. It's not super complicated. All right, so DNA helicase unzips the DNA down the middle like a zipper, okay? I'm assuming that all of you have unzipped something at some point, and you know that you pull the zip down the middle and then you get half the zipper on one side, half the zipper on the other side, okay? And that's exactly what happens in DNA replication. So right now, we're gonna pretend that our hands are DNA helicase, and we're gonna divide our DNA. So here we go. We have our DNA helicase. It goes zoop down the middle. Our DNA is divided. So we're gonna be making a new copy. So you're gonna take this side and you're gonna fold this edge to meet this fold right here. So you're gonna take the edge and you're gonna fold it over so that the edge meets the fold. So you can see, oops, everything right there, okay? And then you're gonna do the same thing on this side. So you're gonna take this edge and you're gonna fold it over to meet this fold. So now we can see our bases. So before, we had two strands of DNA that were connected in the middle. Uh-oh, I'm really bad at folding. With some hydrogen bonds. This DNA helicase, which was our hands, broke those bonds down the middle, and now we have half a strand over here and half a strand over here. Okay, so we have, this is our original DNA, half, and this is half of our original DNA. Right, so that was good. DNA helicase did its job. So now we're going to go to step 2. All right. So step 2 is another enzyme called DNA polymerase. Okay, so it looks like polymerase. Anything that ends in ase is an enzyme. And what this does is it's going to pair the exposed bases with a new complementary, our new word that we learned, complementary nitrogen base. Okay, so we learned that these bases are adenine, thymine, cytosine, and guanine, okay? And this results in 2 exact copies. It's very important that it is exact of the DNA, okay? So let's imagine that you were you're writing these notes, right? If at some point you write down the wrong words or you write them in the wrong order, you're not going to be able to understand what it was that I'm trying to tell you. And then if you don't know what I'm trying to tell you, you're not going to be able to answer questions on your worksheet, and then you're not going to be able to pass the test, and then you're not going to graduate from no, not really, but you see the point. You have to copy it exactly. This DNA, it needs to be exact. So what we're going to do is we're going to list right here our complementary base pairs, and then we're going to actually make our matches, okay? So we're going to title this Complementary Base Pairs. All right, because they are nitrogen bases and they come in pairs. And it always is the same 2. They're couples. They never ever break up. Ever. Even if they break up, they just get another one that's exactly the same. Adenine's like, thymine's the only one for me. And even if they break up, I'll just find another thymine. And it does. Okay? So we're going to have A goes with T. Which also means that T goes with A. C goes with G, which also means that G goes with C. All right? So now we're going to draw our complementary base pairs for both sides of our DNA. All right, so over here, we have an A, so we're going to match that one with a T. Then we have a T, so we're going to match that one with an A. All right, we have another A, so that's going to be a T. C with G, C with G, G with C, T with A, T with A, C with G, A with T, G with C. All right, so now we're going to do the other side. Now the other side's different because we have different bases on this side. So this time we're starting with a T, which means we're going to add an A. We have an A, we're going to add a T. We have a T, going to add an A. G go with C. C goes with G. A goes with T. A goes with T again. G with C. T with A. And then C with G. All right, so now we have the complementary bases added on both sides. So you're going to take your colored pencils. You're going to pause the video, because I'm not going to make you watch me color these in. And you're going to color them the same colors that you did for the originals, okay? Okay, so hopefully you paused and colored, and now your notes look like this. I just colored in circles. But you'll notice now when you look at this side and you look at this side, it's exactly the same. T, A, T, T, A, G, C. We made two copies that are exactly the same letter for letter because we had our original DNA, right? And we unzipped it down the middle, and then we just added new bases to either side, which is pretty genius if you think about it. And your body does this all of the time. All the time. There are so many things that could go wrong, but they don't, because your body has this really great system in place that is very rarely makes a mistake. And even if it does make a mistake, there are actually other molecules that will go and kind of proofread it to make sure nothing happened. And if something did happen, they'll fix it to make sure everything is word for or letter for letter exactly correct. Okay? So from this one strand of DNA, we managed to get two exact copies. Okay? And so now these copies will be divided in the next phase of the cell cycle, which is mitosis. So then we'll do prophase, metaphase, anaphase, telophase, where these two will go to each go to their new home. This one will go to its new home, this one will go to its new home, and everyone will be happy. Okay? Now, I just want you to know that these bases are also attached to a sugar and a phosphate. Okay? They're not just like randomly attached by themselves. I'm just not going to make you draw every single one. So just draw a couple so you know that there is a sugar and a phosphate attached to this, and so that these are exactly the same on this side as it is on this side. Okay, and same over here. All of these sugars and phosphates are all over here too. We're just not going to draw them because we have other things to do. Okay, so I'm going to draw a couple. This is a sugar and a phosphate, and they're all attached, and yay, everyone's happy. We have our backbones, which are these ups and downs. We have our rungs, which are the acrosses. And that is how you replicate DNA. Okay, so again, you're going to take this onto, this is page 48 if you're in Miss DeLuca's class. If you're not in my class, I feel sorry for you. Just kidding. I don't know what page this goes on in your notebook, but in our notebook, this goes on page 48. So you're just going to tape it there, and then you're going to go to the next video, and I will explain to you how to fill in the worksheet. Thanks for watching.